Hello again, everyone. I am now in month 7 of my research project, studying the methylation patterns of ash trees at different ages and severity of ash dieback disease. So having sent my samples for whole genome bisulfite sequencing, I received the sequencing data and began developing an analysis pipeline. And this involved aligning to a reference genome and calling regions that were differentially methylated between samples at different ages and severity of infection. And having never carried out any sequencing analysis before, this was a very steep learning curve for me, and I had to quickly get to grips with software such as Terminal and the R package DMR caller. My bioinformatics skills have therefore improved considerably, and I've gained a greater appreciation of how you can carry out analysis to get patterns out of datasets. So from here, my next steps are to generate graphs from the analysis of differentially methylated regions and begin finding correlations and forming conclusions about the effects of age and disease on ash tree methylation patterns. So another one of my aims was to assess whether characteristic methylation patterns were transgenerational and could be inherited from parent to offspring. And this involved me attempting various germination protocols on ash seeds, which so far have not been successful, and in a few cases where radicals were seen, they did not grow into seedlings when they were planted. I'm therefore trialling a few new methods with different incubation temperatures, and I'm hoping to see germination within the next few months. So overall, whilst I have spent the last few months focusing on the analysis pipeline of my sequencing data, I still feel as though my project is progressing, and I've had the opportunity to develop skills in bioinformatics as well as practical lab skills. And I've really enjoyed finding patterns in the data and discovering how methylation is involved in ash tree resistance to dieback disease.